Hi and good afternoon in Korea. In Stockholm, it's quite early morning, but we are very happy to see you. Today we want to give you a webinar about the biorelevant matrices provided by Biolamina for 2D and 3D culture. And let me see. No, this doesn't want to. Now we go. So I just want to talk a bit. So I think we've been already introduced. So I'm Teresa and the application manager, and this is Noah, the application specialist here at Biolamina, and we're very happy to give you this webinar. For the content of the webinar, we would start first with a bit of introduction also to the company, who we are, and then talk about our product advantages, what they have. And then we go a bit deeper in the actual applications of like hepatocytes and neuronal cells, what has been published there with biolaminins. And in the end, this will be rather a large part. We talk about the third product, which is BioSilk for relevant 3D cell culture. So Biolamina, we believe actually that biorelevance is a key to successful cell culture. And Noah will tell you a bit more about what we mean by biorelevance and what it is that we actually produce here. As I said, we are a company which was founded in Stockholm. We are based still in Stockholm and Sweden. Now we have also a presence in the US, in Boston, but also worldwide. We have in collaborations and customer in over 45 different countries. And yes, our size of the company, I think this number has to be already updated. We are around 60 at the moment already. And yeah, the company was founded on lifelong research of two professors at Karolinska of laminins. And please, Noah, this would be, you can start this one.
Yeah, I start from here. I don't need to share. Teresa mentioned the hive. So our technology is laminins, and laminins, as you might know, are key proteins in the basement membrane that's surrounding almost all the cells, as you can see also in the pictures. And it's very essential for the extracellular matrix, and it's very tissue specific and has a direct binding to the cell surface integrin. And maybe you can, yeah, exactly here. And so it has a role in adhesion, migration, proliferation, and differentiation. And go to next. And so as you can see, that how does a laminin look like? Each laminin, if you look at it, each laminin consists of the 3 intercoiled chains that will be alpha, beta, gamma, and it has the expression pattern changes during development of, and it is very tissue specific, and it provides support of cells, as I talked about it in the previous slide, for migration and cell adhesion. So it's so important. And it has a very unique advantage. It's human recombinant laminins, is animal origin free, chemically defined and xeno-free, and not lot-to-lot variation. And it has a high yield, even without FGF. So laminins are a very large family, and at least 16 different isoforms are expressed in the mammals. And at Biolamina, we have 9 Biolamina isoforms from this 16. And respectively, the laminin isoforms are named according to their chain composition. So as I said, the chain is composed alpha, beta, gamma. So for example, LN111 is going to be alpha 1, beta 1, and gamma 1, and the others also are the same as this structure. And so our technology produces laminins, and we support your cell culture needs, and cells are in continuously interact with the basement membrane, and laminin are a major component of ECM. And to summarize, to say that without the right combination of the laminin isoforms, cells and tissue, they become dysfunctional. So in-body laminins are niche-specific, and almost all cells in the human body grow on a specific laminins. And so as you can see also that we have the human body here, and we have the different isoforms of the laminins, because laminins are very niche-specific. And our laminins are robustness, high quality, means reproducible results, biorelevance, niche recap means functional accuracy, and easy handling, operator and instrument independence, and that can save time. Go to the next slide. Yeah, so as I said, laminins are niche specific, and here are an example. So here that we have the neural cells that are recommended laminins would be LN111, LN211, LN121. The next picture is the lung, and for the lung cells, our recommendation would be LN111, LN521. For the eye cells, LN521, LN511, and LN332. And here is the cardiomyocyte cells from the heart cells. Would be LN221, LN521, LN221, and skin cells LN511, LN521. And here the last is the endothelial cells that our recommended product would be LN411 and LN511. Good. Yeah. So one of our very important laminin is Biolamina 521 that support the human pluripotent stem cells. And here at this slide, you can see that Biolamina 521 supports an easy human pluripotent stem cells tenfold proliferation every 4 days without any spontaneous differentiation. So if you look at the morphology of the cells from day 0 to day 4, so they grow near confluence at the day 4, and they are ready to passage with or without the enzyme. And if we look at the staining, immunofluorescence staining, you can see that when the cells are cultured on LN521, they remain pluripotent with OX4 staining, that's the pink one, and no area of differentiation. And here is the LN521 compared to the other matrices. So that's and the cells that they are cultured on LN521, they are high pluripotent stem cell survival on LN521 to compare to the other matrices. Cells that they are cultured on LN521, they, as we showed you in the previous slide, they are pluripotent and they are genetically stable on LN521 after the long-term passaging. So at the first, you can see the genetic stability that more than 20 different human pluripotent stem cells are cultured on LN521. And yeah, we have a genetic G-banding and SNP arrays that shows the genetic stability after long-term passages. And in terms of the pluripotency, there are homogeneous expression of the pluripotency marker proteins from the, if you look at here, that's the HE stained of paraffin section of teratoma that shows differentiating of the tissue lineages. As you can see, NE is a neuroectodermal, EN is endodermal, and CA is cartilage that indicating mesodermal. So all three germ-layer differentiate. And here is the FACS analysis after 10 passages on LN521 for pluripotency marker OX4 and SSE A4. LM521 is the key protein to rescue of alpha-5 knockout stem cells. So when we cultured the cells on LM521 and the other matrices for the cells that they don't have the alpha-5, the cells only survive on LM521 because LM521 is the key protein to rescue of the alpha-5 of the knockout stem cells. And you can see it here that the cells are here survived and rescued to compare to the other matrices. And so cells die on other substrates upon an LM521 knockout, and only exogenous alpha-521 can rescue the knockout cells. And as you can see also here, yeah, that only the cells survived more on the one that was cultured on the LM521. And we have different isoforms for different applications that will help you to find the biorelevant laminin that is better suits your experiment objective, that Teresa will go through the applications.
Thank you, Noah.
Thank you.
So as you heard, the first part was a lot more about human pluripotent stem cells. This is where we have a large expertise in. Can be both human embryonic stem cells as well as human iPS cells. But of course, this is only one part of cell culture, and the next part would be the desired differentiation of those cells, and we picked here, as I mentioned in the beginning, two differentiation directions. So one is the hepatic, which I want to speak about first, and the other one is the neuronal. And I want to just show you some case studies and papers on it. I see, I have not anticipated that. So the first ones I want to talk about is biolaminins and hepatic applications. So right now, this is quite a big amount. I picked a overview of the paper, which I right now published with our products in laminin differentiation. And I want to specifically talk about, highlight 3 different publications out of those ones. And I will first start with the differentiation of human embryonic stem cells into hepatocytes. So in this publication by Cameron et al. From 2016, they compared human embryonic derived hepatocytes, which were differentiated on either laminin 521, laminin 111, and they compared this to the culture matrigel. And what they saw, especially in the later culture days, like at day 18, but also already early, that key hepatic proteins like albumin or CYP3, they have up to, I mean, for albumin, this was almost a 10,000-fold higher expression when they were cultured, and this is the white bar here, on a laminin 111. Although I must say here in the publication, this is actually a ratio. They call it LN111, but it's a ratio of LN521 and LN111, a mixture, sorry. And this is superior, and this is what Noah already said, that cells can have a very specific tendency, which laminin they like to be cultured on. So you can see that the mixture of LN111 and LN521 is already superior than, like here in this gray bar, than just the culture on LN521, because this laminin here provides them the right culture condition. But not only functionally are they, you can see it here as well, also for CYP3, for the cytochromes, the right culture conditions very much improve the expression of those proteins compared to matrigel, which is the black bar. But also they did actually RNA-seq on those ones, and what they saw is that on the left side, so the yellow, pink, and red ones are the matrigel-derived hepatocytes, and in blue and green are the laminin-derived hepatocyte-like cells compared to the original embryonic stem cells and to primary human hepatocytes. And they saw when cultured on the laminin isoforms, there is a significant decrease of embryonic stem cell genes as well as colon-associated genes, which seems to be quite often still upregulated in the hepatic cell lineage. And all in all, the authors concluded that laminin, in this case, it was laminin 111 mixed with ln 521, actually allows an efficient specification and superior performance of those cells. Another publication I just want to highlight here is from Ong et al. From 2018. Here, the authors compared different ECM proteins, and they created a so-called hepatocyte likeliness index, which is based on different parameters from isolated adult and fetal hepatocytes, their morphology, different protein signatures, and so on. And they compared this index to the cells cultured and which proteins has the highest effect, which ECM protein has the highest effect on this hepatocyte likeliness index. And in their publication, they found out that among a lot of proteins, and in total, I don't have it shown here, but in total they tested 58 proteins, and only 8 proteins shown here had a positive effect. And from those 8, the most positive effect on this created index was the human laminin 111. So Ong et al., what they did, they cultured then cells, either hepatocyte derived from human iPSC cells. In this publication, they call them IHAPs. They differentiated them on laminin 411 and compared them to collagen 1. And I think this you can, so this is the fluorescence staining you have, and I think you can appreciate the really nice staining of albumin and A1AT. And this is just the quantification of the stainings. So the iPSC derived hepatocytes have a much higher expression of the albumin and A1AT protein when cultured on laminin 411 compared to collagen 1. So all in all, actually the culture improved not only cell survival, in here higher fluorescence, but also higher functionality for those cells. A third publication I want to highlight when differentiating human pluripotent stem cells into hepatocytes is a publication by Mesequer Ripoles really recently, just from last year. And I don't want to go too deep into here, but they had shared a very nice graphical abstract. And in this publication, they create hepatocyte spheres, but they 3D models, and used LN521 to differentiate into hepatocytes, endothelial cells, and hepatic stellate cells. Then those spheres are formed by self-assembly into specific well plates. And in the end, the authors created an automated defined process, which will, because it is quite cost effective this way to scale up and reduce the variability. This is what Noah mentioned. We don't have, with laminins, you don't get the batch-to-batch variability. So with the reduced sphere variability, you also have a better cost effectiveness. And all in all, this protocol they developed proved a very good tool for liver disease modeling and drug screening. In the last part of the liver applications, I just want to also mention that there are publications where they used laminin for the culture of primary human hepatocytes. And I want to just highlight a bit the fact that laminins are not only specific in different tissues or different cell types, but it also depends often on the development of the cells. So fetal versus adult might have a different expression. And having a glance on the publication by Watanabe et al. In 2016, they cultured primary human hepatocytes on different laminin, as you can see here, 8 different laminin isoforms, as well as the tumor extract, so something like matrigel or collagen 4. And as you can already, I think, see in this picture, the laminin isoforms give a quite different morphology and self-growth than, for example, something like matrigel. When the authors looked on the functional assays, so they looked on the concentration excreted by the cells of albumin, again, A1AT, cytochromes, and glucose-6-phosphate, they saw that mainly three laminin isoforms are the main isoforms which promote hepatic functions and show the best morphology pattern. And in their case, it was laminin 111, laminin 511, and laminin 521. I want to come quickly back to the publication I already presented by Ong et al. From 2018. They not only derived hepatocytes from iPSCs, so they called them IHAPs, but they also compared those generated IHAPs to primary fetal and adult hepatocytes. And as they found out with this hepatocyte likeliness index, 411 seemed to be a very good matrix for them. They compared the expression of functional correlated genes, and in blue you see the expression of iPSC-derived hepatocytes, which again here is called IHAPs, compared to collagen, and the functional gene expression was mostly significantly higher when cultured on laminin 411 compared in the black bar to collagen. Also, when fetal hepatocytes were cultured on 411, they found the gene expression proved significantly higher of the functional genes compared to collagen. Yes, so all in all, we concluded that if you're using our products for human pluripotent stem cell expansion, especially when you are wanting to differentiate later in the process, we offer 3 different products, which is one, our research grade, the LN521, so this is the isoform we would recommend. And but LN521 as an isoform also comes in a cell therapy grade, which is just produced in different cell line, but allows you to scale up much more. So this is then MX521, or with even more extended documentation and quality documents ready to take it for the cell therapy direction, CT521. If you then want to differentiate the human pluripotent stem cells into hepatocytes, there is an array of isoforms depending on which cells it is exactly, which protocols would it fit you. So then it would be the isoforms of 521, 511, 111, and 411. Or if you're culturing primary human hepatocytes, it is, by the literature, very similar isoforms, which actually a great sign that differentiated cells also prefer the same isoforms as the already primary human cells. And here I just wanted to give an overview again of the publications, which I highlighted some of them now in our talk. The next application section I would like to talk about is the neuronal applications. It is going to be a bit shorter. I wanted to focus here in specific, we have on astrocytes, specifically on the differentiation of human pluripotent stem cells on astrocytes, into astrocytes, sorry. We have at Biolamina, we have a very deep understanding of the neuronal culture and can culture many, have a lot of protocols of differentiating into different cells. I choose astrocytes for today, just by showing certain different ones. So this was a publication by Lundin et al. From 2018. They compared different astrocytes models, which are protocols currently available. And they did a transcriptomic analysis of, for example, commercially iPSC-derived astrocytes, primary astrocytes, and then they developed their own model, which is called NAS-Astro. So this is also human iPSC-derived astroglia cells. And if you can acknowledge here that those cells show actually the least variability and high reproducibility, and each cell line or each NAS as2 cell line was derived from a different donor, and they group, as you can see here, they group already very nicely together. And when the authors did a functional characterization, they saw that the NAS astro model actually has is positively reacting to all of the different analysis patterns. So the transcriptome is correct, the proteome. Then they have some functional assays done with the uptake or inflammatory response. And an astro cell, so their IPC-derived model on LN521. I forgot to mention that, but that is what the protocol was about, to differentiate on LN521. Is the only model which actually has a positive response in all of them, whereas other models, for example, the commercial derived astrocytes, have already problems in some of the functional assays. So these authors actually have a very great protocol published now on an efficient and robust generation of human iPSC derived astroglia cells, and the whole protocol takes about 28 days, starting from iPSC and cultured throughout on the LN521. Another publication I want to highlight here comes from Delsing et al. In 2019. They compared also again human iPSC-derived astroglia cultured either on LN521 or on L2020, which is the Sigma-provided tumor-extracted laminin. So in green, you can see the laminin 5:1 expression, and red is the L2020 expression in different iPSC-derived astrocyte models. So you can see for most of all the marker expression, the LN521 cultured cells prove superior in the expression, have a much increased expression here. And as well, this was not only true for the mRNA expression, but as well for the protein expression of relevant astroglia genes. And they even went a step further and checked if in a co-culture together with endothelial cells, if a culture on LN521 would also improve the blood-brain barrier function, which is a very important function for astroglia cells. And indeed, the expressed markers like cadherin or the Willebrand factor, they all have significantly increased higher expression when cultured together with astrocytes, which were derived on laminin 5:1. So for astrocytes, so far, those are the publications which are published at this point, and the list of our recommended products and isoforms is a bit shorter. So again, mainly we recommend the isoform 5:1, but of course there's always a communication with us and the scientists to see what isoform would fit specifically your model best. In those cases, the publications proves 5:1 to be most effective. And again, we have it in a research grade as well as in 2 different cell therapy grades, depending on which kind of quality documents you might need. And with this, I would like to conclude our 2D cell culture part. So this has been, you have been seeing now the culture of human pluripotent stem cells, as well as the differentiation of those into hepatic and neural, specifically astroglia cells, mainly on a 2D model, although I presented one 3D liver model as well. Now I would like to head into the 3D application, where we have one very specific, very unique product. And the product is called Biosilk. As you can see, I already put the spider up here. So it is mimicking a natural spider silk protein. And a natural spider silk protein looks schematically like this, with an N and a C terminal domain and an array of repetitive units in between. So we produce Biosilk recombinantly in E. coli. So in the production, it's just not possible to have this long, very long repetitive units. So the units are a bit shorter, but we have the same C-terminal domain. The repetitive units are exactly the same as in the silk protein. And the biosilk is also functionalized with an RGD domain. And the RGD domain is actually a cell binding domain. So a lot of like fibronectin, vitronectin. This is a domain which is very known in those proteins. So it just enhances the cell binding additionally to the biosilk. When biosilk is compared, for example, it was done, by Johansson in 2019, biosilk has a different appearance than, for example, a hydrogel has. So biosilk, you have the silk fibers, and you will, in the following slides, hear about what is the advantage of having rather fibers than a dense gel. But I start here by showing the image from the publication where they compared biosilk to a hydrogel. In this case, it was alginate. And I think what is first very visible is that on biosilk, the cells proliferate and migrate much faster. So on day 7, you can see the field is completely confluent with cells, whereas in the hydrogel, cells haven't been proliferating as much. Also, I think you have to acknowledge the cell shape. So on the biosilk, the cells maintain their focal points. They maintain a more spindle-like extended cell shape, whereas on the hydrogel, cells often have a more rounded morphology. And of course, we prefer the spindle-like, what is more physiology found in the body. And the scientists in this paper concluded actually for the reason why that is, that on the biosilk fibers, cells can adhere with many more integrin pairs. This is the network you see here, with many more integrin pairs than they can in the hydrogel. And that's the reason for this advanced proliferation and migration along those fibers. I want to share one of the absolute most recent publications. I will talk about this also a bit later. So by Sozzi et al. Just published 2 weeks ago, actually. So they did an electron microscopy image of the biosilk when cultured human pluripotent stem cells on there. And as you can see the fiber structure is more like a foam-like network, and the cells which can then grow and migrate along the fibers. And this again is on day 7, where the cells, you can see how nicely they form an attachment with the fibers. So biosilk has been proven an ideal substrate for the proliferation of the human pluripotent stem cells. And next is this is how it would look, obviously, in an electron microscope. This is how the silk would look underneath a fluorescent microscope. So you can acknowledge all those fibers, all the cavities formed. And about this, I want to talk a bit in the next slide. But the assembly process, as you can see, the cells are really nicely integrated and very nicely spread. I think this was already seen here, but very nicely spread across the fibers. There's no so denser areas. And also we have, and I just wanted to highlight this here, biosilk can be also used to already co-culture different cell types as well. And it allows, for example, here it was for vessel formation by endothelial cells, which were cultured together with skeletal muscle satellite cells, because the silk allows the cells to actually self-organize in the respective network. The co-culture is actually a very effective method here. When you see this silk network, you can see that it is like a foam, rather. You have larger cavities, and this is actually very much acknowledged in the literature already by several publications. I highlight those two now. So these are the cells stained by DAPI, and you can see you have those porous, we want to call them microporous structures or cavities or whatever names you want to come up here, but they have a very, very important function. And one of the functions was highlighted in the paper by Fiorenzano et al. That is the distribution of oxygen and nutrients. And that is visualized by, they compared a conventional, and this was a matrix gel organoid, to a silk organoid. And after a culture of 6 months, it's very much known that in hydrogel organoids like matrix gel, you develop, or the organoids develop a necrotic core. And the necrotic core is often because the nutrients and oxygen cannot come, don't have easy access to the core of the organoids. And they've done a tunnel staining, so you can see the apoptotic cells really clustering here in the middle. But in the biosilk, you don't find as many apoptotic cells. And you can already see that here, you have much more of those porous structures, of those cavities, those ones here, and they allow for oxygen and nutrients to penetrate the organoid all the way through. So all cells are exposed to the same nutrients in the media. And I want to highlight also later on when I speak a bit more deeply about the biosilk applications. But this is a main feature of the biosilk, and in the latest publication, they also measured actually the expression of apoptosis, hypoxia, and necrotic pathways. And in the biosilk, compared to non-silk hydrogel, you always have a significant lower expression of those apoptotic, hypoxic, and necroptotic pathways. And as I said, therefore, the biosilk can be used to culture quite extensively, up to 6 months, actually. Another thing I already want to highlight, because this is often spoken about, a conventional silk organoid will have a very round shape. This is just due to the nature of the gel. In biosilk, you won't see that. You will see quite a less roundness. It will look more a jagged edge. Also, when they looked on the roundness ratio, it's significantly lower. However, I want to say that, so you see, they might not look like the organoids you're used to, but both scientists, Fiorenzano et al. And Sozzi et al., RNA-seeked the different organoids developed on biosilk, and here I need to explain. Both publications are for brain organoids, which I want to dive a bit deeper in a second. And they compared conventional, like the hydrogel, like matrix gel organoids with pure biosilk proteins, organoids, sorry. But also, and I want to talk about this later, biosilk can be compared with any of our laminins to give you a better specification for the cells. So biosilk compared with a laminin specifically for brain development, which is LN111, they compared the gene sequencing of all those. And as you can see on a conventional hydrogel organoid, you find suddenly some organoids express or have different cell clusters and different cell type composition, and they vary also a lot between the organoids. And the variation and the desired cell type actually increases with the biosilk and LN111 combination. So not only do you get, so first your variability lowers because both biosilk and LN111 are defined and xeno-free, so the variability lowers, but also you get more of your desired cell type. And the same was shown in Sozzi et al. That again, it was a brain organoid. So they compared different markers, for example, pluripotency markers, neuroectoderm and mesoderm, as well as endoderm. Compared on the left side is the non-silk hydrogel, on the right side is the silk protein. And you can see when you differentiate, of course, so this was after differentiation, you want to have a downregulation of the pluripotent genes, which they saw exactly in the biosilk laminin proteins. And the conventional organoids still have quite some upregulation of some pluripotent genes, which is of course not desired. And as well, the BioSilk was much better in the upregulation. They saw much better upregulation of the neuroectoderm genes, which were exactly the ones they wanted to be differentiating into. I can see that our time is running a bit short, so I maybe just go through this quickly. So there are now 3 publications specifically for neuronal 3D BioSilk organoids. However, BioSilk can also be used in many others. I think I'm just gonna very quickly go through this now. I just want to highlight very quickly maybe here that what I said, that the nutrients can reach the core, and this doesn't not only lower the apoptosis and necrotic in the core, but also actually the functionality. So here they checked the specifically dopaminergic neurons, and the functionality or the expression of those specific markers are much more upregulated in silk proteins. So here, in organoids here in green, made of BioSilk and laminin 111. Cells, as I said, can arrange, can organize themselves within the BioSilk. So this is the conventional silk, this is the BioSilk, and as you can see, usually, and that's a bit, because of the necrotic core, you would only see the neural rosettes on the outside in conventional organoids. However, in the silk, you can see them throughout the whole organoid because the cells have the access to the oxygen and the nutrients and are much better in migrating along the fibers. As well, they measured the functionality, so the action potential, but also the ion currents. And as you may know, in the conventional organoids, you cannot measure quite from the core due to, as I said now many times, the apoptotic cells. However, in the BioSilk laminin 111 organoids, the core is not as necrotic or barely. So you can measure also from the core and get really nice measurements. So wherever you measure, you don't have to worry which cell are you taking, because all the cells will have an absolute similar functionality. Just want to highlight that on BioSilk, as I said, although we speak a lot about the neuronal cell type, a lot of different cell types have been tested on BioSilk and are shown to work beautifully with it. We have already quite a range of publications published for the BioSilk, either for directly BioSilk 111 or BioSilk 5:1, so BioSilk combined with laminin 5:1, but also just for pure BioSilk, which can be then, of course, more adjusted. As I said, we have 2 different products here. One is BioSilk 5:1, which is combined BioSilk, combines already with laminin 5:1, or to completely customize your cell culture, you can combine BioSilk with any of our laminin isoforms. So whatever isoform proves best in your culture, you can combine it with BioSilk to create a 3D organoid. And the BioSilk 5:1 product is, this one is just 1 extra pipetting step. So it's not so much more than when you purchase directly BioSilk 5:1. I want to quickly speak about the protocol, but this is also something we can, of course, send out. The protocol is very variable at this point. To create those microcavities, you mix BioSilk, laminins, and your cell suspension in a droplet, and this droplet is foamed up in a dish, stabilized, and then it can be cultured. As we said, we have publications culturing up to 6 and 9 months. However, the silk can be also more used as a sort of a coating. After a while, the silk can be also there are different methods. We need to talk about this more closely, but we have also protocols where the organoids are directly free floating. This protocol is where it is attached, but also attached protocols can be loosened later from the bottom and have a free floating phenotype after, let's say, 14 days. It really depends. We really can customize there how your usual differentiation protocol looks like. And this is how the silk in the beginning, when you create it, looks in the beginning. So this is the BioSilk mixed with LN, for example, LN 5:1 or LN 1:1, and your cell suspension. This is how it looks foamed. And then over the course of 3 days, the bubbles disperse a bit. This dome shape goes a bit smaller, but in the end, you're left with a beautiful silk fiber structure, which looks like this in the microscope. And the cells will then self-organize and migrate along those lines. As I said, our protocol is very, very variable, so that's very good if you reach out, ask specific questions, specifically if you have different ideas about culturing on 96 wells versus 24 wells, or if you think you want the organoid to be attached to the bottom or free floating, or first attached and then free floating. There's a lot of variabilities we can adhere to. But for the most classic protocol, which is the attached organoid, which can be then released later on 24 wells, we also have a very nice recorded session, so it's very easy to you for following the protocol. And with this, I want to very much thank you here, both from mine and Noah's side. And we are very happy that we could have this webinar with you, and we are of course open to any questions at this point. Thank you.